I'll be covering spatial sequencing with the nanostring GeoMx. And this is what the nanostring GeoMx actually looks like in the MoMA lab. But of course, machines doesn't do it alone. So here we have the GeoMx lab team. That besides me is Sarah Neville, who's been responsible for setting up the lab, implementing the method and so on. And then Jesper in the background is part of the histo team, and he's helping Sarah out to prepare the samples. And then, of course, Morten is responsible for the bioinformatics, and he'll cover that in a little while. What I'll cover is to talk about the GeoMx principles. I'll tell you what assays are available, a bit about study design, and then I'll show you some real-life examples with images from the GeoMx. What can you then use the GeoMx for? Well, basically, you can use it to spatial profile any tissue you can get to stick on a regular plus glass slide to see here. So you can look at the core needle biopsies. You can look at the FFPE tissue blocks, tissue microarrays, slide-mounted tissue, fresh frozen tissue, and even cells. If you can get them to stick to the glass slide, then we can look at them. What's important to understand about the assay is that you actually need two different types of reagents. First up, you need the imaging reagents, or as we call them, morphological markers. And these are used to actually image the cells that you're interested in. So these can be actually conjugated antibodies, or they can be RNA-scope probes that are also fluorescently labeled. So we can use up to 4 different markers. And use these markers to select the cells that you want to profile. And what do you want to profile them with? Well, that is your profiling reagents that you see down here with the blue caps on. And they can be either antibodies as well or RNA probes. So you prepare your slides in the lab. It takes between 2 to 4 days, depending on which assay you use. And then once this has been done in the lab, you're actually ready to use the GeoMx instrument, meaning that you are ready to go to the instrument. So the high-plex mixtures of profiling reagents means that this is what actually generates your data. And again, they can be either proteins, and you can do up to 140 plex proteins at a time, or you can choose RNA reagents. And here you can look at the entire transcriptome with more than 18,000 probes for the human assay. And you can also combine them so that you look at both proteins and RNAs on the same run. It'll cost you a little bit of sensitivity on both proteins and RNAs. So that's, of course, depending on what you want to look at. But the sort of readout from both the protein and RNA is this DSP barcode, digital spatial profiling barcode. So you do not actually sequence the RNA directly. You sequence this barcode that contains a tag for the target you're interested in. It contains a unique molecular identifier and then some primer sites. So this is the actual molecule you take downstream. And it is attached to either the antibody or the target, the RNA probe, with a photocleavable linker. Then you can cleave off using UV light. So again, we go back to the lab. We prepare our samples or slides with either the RNA probes or the antibodies containing our tags. Then we add our fluorescent antibodies or RNA-scope probes in order for us to actually see what is on the slide, our morphology markers. And then it is actually time for the most crucial part of this experiment, and that is selecting your regions of interest, or as we call them, ROIs. So these are the regions of the tissue you actually get data from. So you do not scan the entire image and then get data from the entire image. You select your regions of interest, and then we can collect from these. And this actually takes quite a while. I mean, we can help you with the software, but we don't know all tissue types, and we don't know which areas you are especially interested in. So this is a collaborative effort choosing the ROIs. And once these have been chosen, then we are ready to UV cleave the lights, and this, or the probes, this all happens on the GeoMx machine. So once the ROIs has been selected, we take each ROI at a time, shine UV light, and then thereby cleave off these barcodes. Then they're collected into a 96-well plate, one at a time. Of course, repeated until we have collected all probes from all ROIs. And then we're actually ready to do our downstream analysis, meaning taking the 96-well plate, doing sequencing libraries, and then doing sequencing on one of the Illumina machines that we have. And then Morten takes over and takes care of the bioinformatic analysis. So these are the basic principles of the GeoMx system. But as I said, the ROIs are sort of the most important step of this process. So you can, of course, choose to select tags from the entire ROI, but you can also choose to subdivide the ROI into different segments. So in this example, in the middle here, we have a cancer specimen, and then the yellow cells here are the cancer cells, and then we can, or the GeoMx can help you create a mask that actually select all of the yellow cells into one well in your 96-well plate, and then the tumor microenvironment can go in another well. Or you can also choose only to take the purple cells, because the way the UV lights are actually used is through these digital micromirror chips. And they have a resolution of 1 micron. So you can actually select individual cells, but you need multiple cells to get enough signal. So usually you will take a ROI and divide it into 2 or 3 segments, and then taking all the cells of a specific type into one well. This, of course, means that instead of having a mixed, in this example, tumor and microenvironment signal, you can divide them into tumor cells only and microenvironment cells only, and then get individual expression profiles for these 2 segments of this ROI. So what assays are available? Well, for human assays, there is the Human Whole Transcriptome Atlas, covering more than 18,000 genes. There's only 1 probe per gene, whereas if you go for the smaller assays, here we have the Human Cancer Transcriptome Atlas, there's 1,800 genes, and then there are 5 probes per gene in this assay. And then finally, you can go to smaller panels as well. So that's for humans. If you want to do mouse, then there's a Mouse Whole Transcriptome Atlas as well, with more than 20,000 genes. And NanoString just released a canine cancer atlas as well, with around 2,000 genes. And then you can actually also design your own custom panels. If you have a specific species you're interested in, then it is possible to design a panel for that. But you can also actually design add-on panels or add-on content to the existing panels. If you're interested in non-coding RNAs, circular RNAs, or whatnot, then you can add them to the existing panels to get more content and more information about your samples. The way the protein setup works is that it is available for both mouse and human. So you have to have this protein core that contains CD45, Ki67, CD31, and then for mouse GFP and for human beta2 microglobulin, along with some controls, both positive and negative. And then you can actually add to this protein core with modules. And these modules are basically divided into 2 main topics. There's one for neuroscience, covering Alzheimer's, Parkinson's, glial cells, and more. And there's also immune oncology protein panels covering, of course, different immune cells, but also a pan-tumor panel and different signaling pathways. And each of these panels is around 10 antibodies. And as I said, you can combine up to 140 antibodies in one run. So you can take nearly all of them and combine them on the same slide. And as for the RNA, you can also add custom content to this. So if you want to know more what specific proteins these panels contains, then you can go to the NanoString website and look them up. Okay. But if you're interested in running a GeoMx experiments, then there are certain considerations that you should have before starting. Of course, you need to know which tissue type to use, and then depending on the age of the sample, of course, the newer the better, but a lot of experiments have been run worldwide with old FFPE samples as well, so it is doable. The size of the RNA probes are between 35 and 50 bases. So it can accommodate really degraded RNA as well. Then you need to figure out whether you want to use fresh frozen, FFPE, fixed frozen, what kind of tissue preparation. And then most importantly, you are going to find out which cell types to investigate. And again, how to find these cells, I mean, which morphological markers do you want to select them? Is that antibodies, is it RNA-scope probes, or how do you want to select your cells of interest? Then, of course, you need to figure out which assay, which profiling reagent. And if you want to go for the full transcriptome, then NanoString recommends that you have at least 100 cells per segment. And again, the segments are either the cancer cells, stromal cells, or whatever is in your ROI. For protein, you can go down to 20 cells per segment. But again, the number of cells also depend on the cell type, the cell size, how RNA-rich or protein-rich your cells are. So you can go lower than this, but it'll probably come at a price of detecting fewer transcripts in each ROI. Finally, money is always an issue. So you need to find out how many slides you want. And as the most expensive part of this experiment is actually the probes or the profiling reagents, and they have a price per slide, then you need to figure out how many tissue sections you can actually put into one slide. And I'll show you examples just after this slide. You also need to figure out how many ROIs do you want to select, and again, how many segments per ROI do you want as well. So, as I said, you can put the tissue on a glass slide, but not the entire slide can be used. You have to put your tissue within this green square that is 3.4 centimeters by 1.4 centimeters. And you can actually fit quite a bit of tissue in there if you're skilled at this. So I have a few examples here. One is from Mike and Bøge's experiment with brain sections. So here we have 4 rather large brain sections on one slide. Up here we have a case from dermatology, Thomas Emanuel. He had smaller biopsies, so he managed to fit 10 into one slide. And then finally, you can also go for the tissue microarrays. So this is Trine from MoMA's bladder group. And there's around 100 cores on this one slide. So you can really get a lot of information from different patients using GeoMx. Then you need to select your morphology markers. As I said, you can have up to 4 different markers, one of them usually being a nuclear stain. So 3 markers for individual cell types. But there needs to be in different channels with different fluorescent signals. So you also need to be able to find the right markers. And usually people have sort of optimized this on their own. And then what we do when we get the samples is that we do what we call a dry run, meaning that we do a run in the lab, but without the expensive profiling reagents. We just add the antibodies or RNA-scope probes, and then scan it in the GeoMx, and then we can see whether it's actually possible to select the cells that you're interested in. And then it's finally time to select the ROIs. And this can be done in different ways. First up, you need to draw some sort of geometric shape. You can draw a circle or a square, or just draw by hand. And again, you can collect all the cells within the ROI, or you can segment into different sections with different cell types. It is also possible to pick individual cells, but again, you need around 100 to do the full RNA assay. There's more sophisticated stuff coming in the next software version, NanoString Promises, where you can do more contours and a more gridded approach as well. An example provided by NanoString, this is colon tissue, where mast cells are here stained in red, and they're dispersed around the tissue. But as I said, the resolution is actually enough for single cell collection. You still need to collect all of these cells within one well to get enough signal, but you can pick them individually and then have them in the same well and then get signal, even though they're not clustered together, the cells. So these were examples of how to collect ROIs and how we haven't run that many experiments, I must say, but these are the experiments that we have run or are about to run. And then finally, I just want to say that if you're interested in looking at more data and more examples, Then at NanoString's webpage, there's this NanoString Spatial Atlas, and they actually have these different brain, lymph node, pancreas, and so on for human, and then for mouse, the brain and the embryo. So if you click, for example, a colon, it'll take you to a page where you can see where the individual rows were actually collected. You can see the different tissue types up here, and then they will provide you with examples of clustering, heat mapping, how many genes you should find in each of these different cell types, etc.